morning, viewer. Welcome to our Glory Globe TV. Here's a really amazing news for our viewer. Yeah, for decades, large stretches of human DNA were dismissed as junk and considered to serve no real purpose. In a new study published in Cell Genomics, researchers at Lund University in Sweden show that the repetitive part of the human genome plays an active role during early brain development and may also be relevant for understanding brain diseases. DNA carries the complete set of instructions and organisms needs to develop and survive, but only about 1.5% of it consists of protein coding genes that determine traits such as eye color, height, and hair type. The other 98.5%, once written off as junk DNA, is now increasingly recognized as an important part of our genome that controls when and where genes are switched on, influencing development, cellular processes, and even human evolution. At Lund University, researchers have been exploring this overlooked portion of the genome. Their latest study shows how specific sequences within the non-coding genome help shape and developing human brain. An underlying question in my lab is how did the human brain become human? Says Johan Jacobson, professor at the Department of Experimental Medical Science and head of the Laboratory of Molecular Neurosciences. We want to know which parts of the genome contribute to uniquely human function and how this connects to brain disorders. In the new study, Jacobson and his team, together with collaborators at the University of Copenhagen, the University of Cambridge, and New York University, investigated segments of repetitive DNA sequences called transposable elements, sometimes described as jumping genes, and these sequences can move around within genome, making them challenging to study. Using induced pluripotent stem cell and brain organoids, miniature simplified versions of the human brain grown in the lab, the researchers studied one particular family of transposable elements known as line 1 transposons. By combining CRISPR gene editing technology with advanced sequencing methods, they were able to switch these sequences off and observe the effects. We have this exclusive report, Glory Globe TV, Delhi, Bharat.